Hi, hello. Well, my name is Chema Martin. I'm a senior lecturer at Queen Mary University of London, and I wanted to thank the organizers for the opportunity to present our work on the sequencing the genome of the bone-eating worm Osedax frankpressi. Annelids are quite a diverse invertebrate group that has evolved multiple times bacterial symbiosis to meet nutritional requirements that have allowed them to colonize many of the most extreme environments on Earth. Like, for instance, deep sea and hydrothermal vents, like you can see here in the picture, Riftia pachyptila, that is the iconic giant tube worm. Among the symbiotic annelids, we have one particular lineage that is Osedax worms, the bone-eating worms or the zombie worms, that have in particular developed a nutritional bacterial symbiosis that allow them to feed exclusively on sunken and decaying vertebrate bones, which is a really fascinating and unique adaptation in the animal kingdom. Actually, these bone-eating worms and the deep-sea hydrothermal vent worms belong to the same group of annelids that we call Siboglynidae. The deep-sea hydrothermal vent worms belong to the group Vestimentifera, and together with Osedax, Scleroninum, and Frenulata, they form this large, diverse group of annelids, symbiotic annelids that we call Siboglynidae. They live, as I said, in different environments, and they establish all of them bacterial symbiosis that exploit different nutritional sources, from vertebrate bones, as I said with Osedax, to the use of inorganic compounds like sulfur and methane to produce organic compounds. Therefore, they also exhibit diversity in the kind of nutritional interactions that they establish with the bacteria, from chemolithoautotrophy, like frenulates, vestimentiferans, and sclerorinum, to the typical heterotrophy that Osedax established with the bacteria to feed and digest and exploit the decaying vertebrate bones. Therefore, when we compare all these diversities, we can infer that the symbiotic association with a bacteria, probably of a chemolithoautotrophic relationship, is ancestral to Siboglynidae, and therefore, and at some point in evolution, the heterotrophic interaction with bacteria to exploit sunken vertebrate bones evolved in the lineage leading to Osedax. However, the fact that we only have genomic resources for lineages within Vestimentifera, like for instance Labellibrachia, Riftia, or Parascarpia, makes very difficult to understand how this symbiotic association evolved initially and ancestrally in Siboglynidae, and how it later on diversified to establish a heterotrophic association in the lineage leading to Osedax. So that's why we decided to face these two different questions by sequencing the genome of Osedax, in particular the species Osedax frankpressi, and compare it with other genomes within Vestimentifera. And this is what I will briefly show in the following slides. So the first interesting thing is that the genome of Osedax frankpressi is way smaller, shorter than those already sequenced and assembled for Vestimentiferans. So it basically is between half and 4 times smaller than other Vestimentiferan genomes. And this smaller size is basically not due to a lack of transcription of transposable elements, because actually Osedax has similar proportion than Riftia pachyptila, but to a clearly reduced gene repertoire. In fact, when we compare the gene repertoire of Osedax, Vestimentiferans, and many other bilaterian genomes, what we see is that Osedax frankpressi belongs to a group of highly divergent, or the repertoire of Osedax frankpressi is very similar to highly divergent annelid genomes, like for instance leeches. And Vestimentiferans also have their own very specific gene repertoire. In fact, when we analyze the patterns of gene gains and losses for Siboglynids, for Osedax and Vestimentifera, and compare it with other annelids, what we see is that there are 2 or 3 actually major events of gene gain and loss in the evolution of this major symbiotic annelid group. There was an ancestral major event of loss that is common to both Osedax and Vestimentifera, and then Osedax kept losing genes at a really high rate. So it's actually the most gene-lossy genome for an annelid sequence today. While Vestimentifera, on the other hand, experimented a common event of gene innovation and expansion in the last common ancestor. So we next decided to focus on understanding what these losses and expansions might mean for the evolution of symbiosis in Osedax. And what we did as well was sequencing the full genome of the symbiont. And what we found is that this genome, compared to those of the symbionts and the Vestimentiferans, has a more versatile and diverse metabolic repertoire. And this clearly complements the gene losses that Osedax has experienced in its metabolic complement. So we identified that contrary to asymbiotic annelids and Vestimentifera, Osedax has lost the capacity to produce 3 amino acids that are proline, arginine, and serine, which are quite abundant, especially proline and serine in collagen, that is the main component of bone. And the loss of the biosynthetic capability for arginine is actually quite interesting because it implies that Osedax and the symbiont are probably in a highly in a high concentration of ammonium inside their cytoplasm. They still have, Osedax has still the capacity to transform arginine into ornithine, because ornithine is important for the synthesis of polyamines, but is incapable of recycled nitrogen. And this probably favors the synthesis of other amino acids that are not present in high quantity within the collagen. We also identified massive expansions of collagenases, of matrix metalloproteases in Osedax, which again makes sense because they have very different domain composition, and they are probably quite broad and diverse to cleave all extracellular matrix of the bone. They are again similar to the enzymes that are involved in synthesis of amino acids. They are expressed highly in the roots, or more expressed in the tissue that is in contact with the bone than in the other part of the worm. We also identified quite interesting losses in Osedax, like for instance, a very reduced immune repertoire, that is the genes that are involved in identifying and keeping control, keeping the endosymbionts under control, especially when compared with Vestimentiferans, which have an expanded and very diverse repertoire of immune genes. And we also identified the loss of a key DNA repair toolkit in Osedax, which actually could be related to the fact that they have this smaller genome size. Altogether, this analysis of the genome of Osedax frankpressi allow us to basically define a scenario of how host and the symbiont interact to digest the vertebrate bone, and also highlighted the fact that similar annelids, symbiotic annelids, with different nutritional interactions, experience different pathways and different routes of genome evolution during while establishing these bacterial interactions. So with this, I would like to thank the people that conducted the work, in particular Giacomo, that is one of the highlighted with the circle, all the Osedax genome team, and the founders. Thank you, and I'm happy to take any question.